Hello, everyone, and welcome to today's webinar, Next Gen Lab Informatics Part 1, Transforming Research with New ELN Technologies. I am Antonina Salcido of Labroots, and I will be your moderator for today's event. Today's webinar is presented by Labroots and brought to you by BIOVIA. To learn more, please visit 3ds.com/BIOVIA. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Stephen Hayward. Stephen has a PhD in chemical engineering with a background in laboratory informatics, chemical modeling, and environmental analysis. For a complete biography of today's speaker, click their name in the presenter's window at the top right of your screen. Stephen, welcome. You may now begin your presentation.
Great. Thank you so much. Thank you for the introduction. It's a pleasure to be speaking with you here today. As you may be aware, this is the first in a three-part series that's discussing the next generation of lab informatics technologies and solutions. So today we're going to focus on early stage preclinical research and the most common informatics solutions that are used there. And then through the rest of this year with Labroots, we'll be hosting sessions focusing on areas such as process development and quality control. So keep an eye out as those sessions are announced. So as I start my presentation here, I'm going to move on to the slides, and I'm also going to turn my camera off just to make sure that we don't run into any bandwidth issues while I'm presenting. So let's dive into our subject. We all know that labs are under constant pressure to deliver products and analysis and results of high performance and quality as quickly and cost efficiently as possible. But too often the lab becomes a bottleneck for projects. They get hindered by inefficiency and there's lots of pressure put on the scientists as well. And why is that? Well, in broad strokes, it's commonly because of a lack of available resources, whether that's because of budgets, a lack of available personnel, lack of resources, or even just due to issues with technology in the lab. There's still too many non-value-added tasks that are taking up scientists' time each day as they search for previous work, they try to collaborate with colleagues in different locations, and deal with project backlogs. And so if they can't find the relevant information that they need, or worse, if they don't trust the data, then they have to spend time reproducing the work as well. And so not only does that waste time, but it wastes resources and increase costs. We've seen that up to 45% of R&D materials are actually ultimately wasted in these extra tasks that need to be done. And so anything that we can do to reduce this helps increase your lab sustainability as well. But ultimately, increasing lab efficiency, it helps ease the pressure points in getting new therapeutics to market faster. I want to focus on lab scientists for a moment because too often we talk about lab informatics in terms of workflows and efficiencies, and it leads us to kind of forget the human factor as well. A scientist that's working in the lab, they have to move between many different systems to complete their routine work. Our lab systems have to serve different roles and personas across the organization, but we also have to consider that scientists perform many different roles in their daily lives. In smaller organizations, one person in a lab could be responsible for all aspects of designing a project and performing all the necessary experimental work and data analysis. At a larger organization, the roles become more specialized, but scientists are still then collaborating with each other in different locations to drive these projects forward. And that takes interacting with many different software applications, not just for the lab work, but for the collaboration as well. Each day, scientists are moving between in-person work and virtual and hybrid workspaces, something that's just been accelerated during the pandemic of the past few years. So scientists, they move between these different dimensions throughout their daily lives, and that can be exhausting to them, and having to move between different types of pieces of software as well each time. So we've talked about the scientists. Now let's talk about what's going on across the organization in these different spaces. So if we look at the therapeutic development spectrum, we could see the different types of activities that occur in each product stream as a new therapy moves to market. So on this slide, if we look at the bottom, the horizontal x-axis, we're looking at the product development life cycle as something moves from early stage research and discovery into preclinical development, and then through clinical testing and manufacturing phases towards being commercialized. And then we have these different product streams that, depending on what type of company you're dealing with, or if it's a large company, it may have all three of them, and even more in some cases. So you have small molecule development. And so in early stage research and discovery, you're dealing with chemical synthesis and screening, and then that moves into chemical manufacturing. If you're looking at a biologics process stream, you may have, yeast or mammalian cell-derived processes, or ones that are more individualized, patient-provided ingredients as you move to smaller patient populations as you move up this vertical y-axis. And then for biologics, you're moving into biologic process development and clinical testing or other biologics process. And so all of those first three ones are delivery to patient populations. But up at the top, now we're moving into very narrow streams where we're talking about cell and gene therapies that are reliant on ingredients from an individual patient. And then you deal with biological processes to produce therapy for them and deliver it back to that same individual patient. So you could see these different types of activities across an organization. And of course, that changes too if you're dealing with medical devices. You would have different types of different domains and organizations in that company too. But if we think about that, in every section, every product stream and every stage of the development life cycle, you have a different lab associated with it, right? As you do the original research and discovery, so you're doing that first experimental work to start with. When you do the development and scale up before you move into clinical testing and manufacturing. And so there are labs at each stage of these processes. And so, they may not be homogeneous either for, each box that we see here. There can be multiple different types with different focuses and specializations. And so, at a large, say, multinational enterprise, you could see how the variety and complexity of these lab organizations multiplies. And that's one of many reasons why we see so many specialized informatics point solutions deployed across the different domains of a company. And so if we think to our scientists who have to navigate these different roles in their lab, we realize how their digital lives could become quickly complex and overwhelming because they have to deal with all of these different point solutions depending on which area they're operating in, and trying to possibly move information from one system to another, or pull it from a different system earlier in the value stream if they need to reference something. So here on the bottom horizontal axis, we have the same product development life cycle going from research through development, QA/QC, and manufacturing. And on the left, on the y-axis, we sort of talk about the different areas of responsibility that these solutions have. So you've got your ELNs that are really concerned with the data and execution, a little bit of management oversight there as well. We move into the LIMS system, Laboratory Information Management System, as you're moving through process development and QA/QC. And then further on towards the product development lifecycle, you have an execution system, maybe electronic batch review. And in manufacturing, you have a manufacturing execution system. And that's not even to touch upon, the product lifecycle management, ERPs, other systems that may be laid on top of this and loosely integrated together in certain situations. But you could see how, it quickly multiplies of something that all the organization has to deal with and the scientists do as well. So a scientist, has to move between these different systems, and sometimes it takes more time to do their work on the computer than it does just to do the work in the lab itself. And then it also makes it difficult to search and find relevant work if it's in these different duplicated systems. Say you may have different ELNs spread across your research organization due to the different types of labs that are using specialized ELNs. And so the only way to find the information is to email somebody and hope they remember where it is, and then they send you the file, and then you have to import it into your system. So it becomes very, complex and overwhelming quickly. And so, like I said, these point solutions have arisen to address the specific needs of specific labs. And that gives these artificial barriers between the systems as well, where there might be overlapping functionality or maybe unaddressed gaps. And then the systems are integrated together in an ad hoc manner to try and simplify the workflows after the fact. But if we really want to simplify scientists' digital lives, we need to eliminate those barriers. And we could focus on developing digital workflows that match the actual lab work and build this out from the start, rather than integrating solutions together that were never originally designed to be joined. So in this way, then, we can streamline workflows for increased efficiency while standardizing the data and procedures underneath for better compliance and overall preserving a single source of truth. So I hope that makes it clear why companies are starting to move away from siloed solutions towards more integrated and platform-based approaches. It's not only for the efficiency and compliance that we discussed, but because when you take a more holistic view of the information generated and transferred across an organization, then you can realize how important it is to have a consistent data model underpinning all of it. And so that's the paradigm shift that we see, is that the data that's driving the healthcare of tomorrow is centered around models where it's possible to create virtual twins of both the products and the processes in your organization. But the way to get there is by connecting all of the dots across that organization, those individual nodes of data, so that we can more meaningfully leverage it in new and exciting ways. So many customers and companies are already using this technology. And probably some of you are customers that are trying to understand things that are going on in your process. And so, the biggest companies that are in the world are already using technologies such as a knowledge graph to better understand their customers and the decisions their customers are taking, what they're interested in, what would better serve them for a new product. And so knowledge graph is something that's a representation of both the quantitative and qualitative facts around your customers and products, and it can uncover new relationships in new ways. And so you see this at Netflix and the algorithm that serves you new information based on what you viewed. Facebook and Twitter are using it. So social networks are doing this type of information so that they can better serve you things in their social algorithms. Amazon obviously uses it to figure out exactly what you need to see when you enter into the website. So there's a lot of applicability here, and we'll talk about how we can apply that into the scientific realm as well. But first, we should describe a little bit what a knowledge graph is so we better understand it. And so if you're not completely familiar with graphical data models, or here on the left, we have a representation of a traditional database, which has the data stored in tables in rows and columns. And generally, traditional databases have a very rigid schema, and they can be difficult to extend. So, if you want to change something in one place, you might have to redo your whole data model. And whether on purpose or by accident, the database might be biased by the individual that comes up with the original design. On the contrary, a knowledge graph is where the pieces of data are connected by edges and nodes, and they're designed to be more flexible, so they're easy to extend and you can continually update them. Knowledge graphs are where we don't let our prejudices direct where the results come from. We have the results and we could follow those nodes and edges and sometimes get surprising conclusions. And that's really the advantage of a knowledge graph, and really what we want to take advantage of in the next generation of lab informatics solutions. So for scientific applications, how do we see this knowledge graph technology helping us to do better science? Well, one of the features of this is that you can combine your actual data that you've run in the lab with data that you've created for modeling and simulations, as well as combining that with data that you can find out on the real world in the internet to infer deeper relationships and enable analytics. For scientists, this sort of technology could help suggest similar experiments. So, if I start drafting an experiment or a plan for an experiment in my electronic lab notebook, based on the information that you're entering and comparing, you compare that to the ontology that's been set up in the system, and it might suggest similar experiments, or maybe even experiments that have already been done. So that could give you a better starting point for your experimental work with analytical methods. And we'll talk about a proof of concept about this in the analytical space in a little bit. You could easily suggest a starting method for a new compound or a material that you needed to develop. And you do the same thing for formulations and so on and so forth. And for the data scientist in your organization, these knowledge graphs provide advanced query abilities. So you could start building those connections between data points that you may not have thought of intuitively out of the box. And you can then combine your experimental data, as I mentioned previously, with data that's available on the internet or in simulations and models. And so all of this is combined to help address the FAIR data schema or similar. It's somewhat of a buzzword in the industry, but it's super important. And I think that's why everyone is talking about it so much. So let's talk about how it all works. Here's a little bit of a primer in terms of how ontology and taxonomy are related to knowledge graphs. Typically, we work with about 10 columns, which is a standard classical hierarchy classification of data. And this is a simple categorization, so there's no cross-domain relationships and etc. You could think of classifying chemical entities, and there you might have organic compounds and inorganic compounds. And underneath organic compounds, you have hydrocarbons, which are then divided into different categories, and so on and so forth, until you get down to an actual structure or entity. And then we get to an ontology where you could start combining those hierarchical relationships. Well, here we're discussing classifying the structures of chemicals, and then over on the right, we're talking about their roles. What are they used for? How did they behave, etc. We have biological roles, chemical roles, and you can see that some of these are safety classifications as well. Some of them are about usage. Has the same material been used in different roles? And you could have many different roles depending on whether you're talking about an analytical chemistry lab, a biology lab, or a standard small molecule chemistry formulations lab. The same material could be used in different places and many different ways. And so when we start combining the taxonomy with the ontology, this is where it gets interesting. This is where the point-to-point relationship that we have in the graphical data models becomes so powerful. And so finally, as I said, adding the knowledge graph capacity, that's when we're actually starting to combine the data with the ontology. So not only do we have the classifications, but we have items that exist in this classification. These edges that the ontology provides to our taxonomy allow us to understand much more about specific subjects and can continually be expanded as we update the ontology or data. So that's all well and good, but how can we apply that to lab informatics solutions? So if we look at an ELN, let's think about what we use it for. We're using it, as you create and perform an experiment. So you're writing down the project ideas and then the experimental results there. Any of the data that you get out of it, whether it's structured or unstructured, that's getting stored in the ELN as well. You need to create the audit trails, and so you're creating proof of the IP there. And you're using it to search and access other knowledge across the organization as well. And so some of that's changing too as vendors expand the capabilities and the integrations for the ELNs. And they're moving towards something that's more of a central knowledge repository and management system. But the question is, can we do more with the data that's stored in the ELN, especially with all of the unstructured data that's generally just sort of written down in free text form and pieces of information that are copied and pasted over? How can we start to extract some value from that? So if you look at what's actually contained in experimental write-ups, there's lots of common concepts and terms there. And so, we have the concept of a study experiment, and experiments can be linked to the material. So an experiment has to use that material, of course. And then the experiment could be used to develop a method, or it could leverage a method that's already in place. The methods are related to equipments and samples, and the methods also generate results, and so on and so forth. And so you could see that the whole laboratory schema can be set out in this fashion in a graphical database that we could take advantage of. So if you could automatically extract and parameterize the data as it's entered into the ELN, that opens up a world of possibilities to use the data in other ways, either immediately or as part of data science initiatives on larger data sets. So one proof of concept that we investigated at BIOVIA to sort of prove that this type of data model can work the way we wanted it to was around chiral HPLC analytical methods. HPLC method development is used in a variety of industries for a variety of purposes. And HPLC method development, for many customers and scientists, they have their starting columns and methods pretty well defined for the material that they're working with. For chiral HPLC, it can be a little bit more of a dark art. And so we wanted to see if we could use data that was available from free source internet data and predict which conditions would be the best to separate several chiral compounds. So in order to do this, we had an open source database of compound structures, as well as HPLC methods that were used to separate those structures. We also had an ontological service that was open source. And then we used BIOVIA Pipeline Pilot to generate some physical constants of the materials that we're investigating to see if there's any relationships there. And then we took all of that information and we put it into our experiment data model, which we call SPR for Studies, Process, and Results. So here's just a brief diagram of sort of how it's all connected. And so up at the top, you could see our chemical classification that we're looking at earlier as a taxonomy. And then we get down to the actual structures. Here we've generated some physical constants of their structures using Pipeline Pilot. And then we're actually connecting the information about the physical constants as well as the structural information with HPLC and method information like the stationary phase, the mobile phase, and all of the conditions that were used to separate those materials. So if we have this all in place, can we use a knowledge graph data model to more quickly develop methods for novel but related chiral compounds? So for the actual process, what it looks like, the way that it works, or that we hope it would work, is that when we identify a new compound of interest, then we set the ontological annotations. So we're giving it the categorization in its physical constants. Then we would find semantically similar known analytes. So, other compounds that play in the same space in this ontology as the compound that we're trying to separate. And then we could hopefully identify the column or columns that will work to separate out those analytes. So for the proof of concept, we wanted to focus just on selecting the right columns rather than the whole development of the method. And then the idea is that once you have that information and you could prove that your data model works, then you can add additional information to the column. And then in the future, when you have new compounds, you're able to go straight to the columns and find out which ones would be able to work to separate out the compounds of interest. So here, we want to select the column, find the analytes that are separated by it, retrieve those attributes of the analytes, and see if there are any commonalities between the ones that we have already and what we're trying to separate out. Then we do the prediction, and then finally add any extra inferred relationships to the column to proceed with method development. So what were the results of this proof of concept? Well, we had a database of 23 chiral HPLC columns and 111 analytes. And our proof of concept was able to infer the correct relationships by suggesting exactly the right set of columns 48% of the time. Well, you might say that's not really interesting at all. I guess if I were guessing that I might be able to do better than that. But we're not talking about a binary choose column A or choose column B. What we're talking about here is actually selecting a set of 5 columns where all of the columns can be used to separate that compound in question. At least 80% of the time, we were able to select at least one correct column that could be used to separate the compound in question. So that's pretty good results. And I think that it's even better when you think about the fact that the individuals who ran this proof of concept within BIOVIA were not in any way chemists. They were just data scientists that just had a basic understanding of how analytical chemistry and HPLC column separations works. So we're really excited about this. I think it's a good proof of how ontology can be used and how powerful knowledge graphs can be in solving scientific problems. We all know that going into the lab and running actual experiments is expensive in terms of materials, scientist time, and in terms of the environment. And so anytime that we can use simulations and models and graphical databases to come up with better starting points, and we have to do less experiments to get our expected and desired results, I think that's a great thing for everybody that's involved. I know I talked earlier about ELNs being the predominant informatics solutions that are found in early stage research. And while that's still true, we're finding more and more that the ELN is becoming a central repository for knowledge across many different types of labs, regardless of what other informatics solutions are in place. So in early stage research, you would use the ELN, to document experimental results and new ideas, how you're testing out different synthesis pathways, et cetera, and testing actual product properties, among many other things. As you move into process development, a LIMS might be the most commonly thought of informatics product, but an ELN is also useful to document the progress of projects. And you could document the validation of synthesis pathways, the outcomes of different test procedures, et cetera, as you move to measure the success of your scale-up efforts. In quality control, there are more rigid testing procedures and batch release tests that are necessary. And so a lab execution system would probably be in place. But an ELN could be used to help manage the workflows and the overall project progress again. And it also helps create the link between commercialization and the initial product development work. And so you could see how it'd be useful to break down the barriers and purpose-built nature of some ELNs and to have something that's more flexible and can be used across the product development spectrum rather than siloed in each area. And that way, then you get to preserve the data in its original format and tie the whole project together as the product moves to market. I want to touch a little bit on evaluating ELNs and moving towards implementing them. And so there's a number of steps that you could take in sort of a progressive manner to make sure that you are choosing the right ELN as you're evaluating it, and how to quickly get the value of it once you start to deploy it. And so once you've sort of launched the selection process, I think the first thing to make sure you do is get the right people involved early. You need people that are, scientists as well as your lab managers. So some of your specific lab users, as well as the ones that are overseeing the labs and understand the pain points that you're trying to address, as well as people from IT, because they're the ones that are involved with the infrastructure and maintenance. And it's at this stage where you want to define the use cases and workflows that you're really trying to address, especially which ones you want to start with before you expand the project to a wider deployment. This is also where you'd identify any barriers to deployment or adoption, and start to identify ways to overcome that. And then make sure that you're communicating early and often to all of the stakeholders that are involved. Then you need to make sure that you assess the requirements carefully for the use cases. So really understanding what your pain points are, which systems you might be augmenting or replacing, because that comes into the scope of it as well. And then what your actual scientists need to succeed. So touching upon the point that I made earlier about, it's to understand the efficiency and to increase the productivity in the lab. It really comes down to what do the scientists need to be supported and be more efficient in the lab. I think it's also important to establish a strategic relationship with your vendor as well. So you're looking to find partners that you can work with to deploy the solutions in a collaborative manner, who are responsive to your needs, but can also help guide you based on their experience and based on best practices. It's also really important to make the most of product demonstrations. So defining the important workflows and capabilities that you're interested in, getting clarity on what features are available out of the box and what needs configuration or customization, looking at the ease of use of the solution and what type of training is necessary, and then exploring further the integrations and configurability of the solution too. You also have to consider the total cost of ownership of the solution. So are you going to deploy it on premises or on the cloud? And obviously lots of people are moving towards cloud first, because you can offload a lot of the infrastructure responsibility onto your solution provider. So you can examine that and also look at what type of validation efforts are necessary, if that's depending on the space that you're working in. I think it's also a great idea if you can get value from reference accounts. If you could talk to previous customers from the vendor and delve into their specific experiences, identify if there are any common pain points and the outcomes from addressing those. You should also ask about ongoing customer support and success, and maybe if they do anything differently as well. And then I think it's also a great idea to try and pilot the system. If you can do that in a small focused lab in an actual production environment, that's a great idea as well, because then you actually have the ability to see how it works in the real world environment, and make sure that your key personnel are involved in testing that system. Be prepared for feedback and to identify key metrics where possible to help measure the value and success. And so if we go back to the sort of informatics landscape slide that I talked about earlier, this is where I want to start to introduce you a little bit to BIOVIA One Lab, which is our solution for removing those barriers across these different parts of the organization, and bringing key capabilities together, being able to manage both the documentation and the knowledge that's being generated across the different labs, to put task planning and procedure execution together, along with inventory management and equipment integration and management, and have all of those together in one more integrated solution where you don't have those artificial barriers in place. So for us, as I said, it's called BIOVIA One Lab. It's a role-based, unified lab informatics solution that can be deployed across research, development, and quality control to help manage and execute tasks, to manage the associated resources, methods, and processes, to aggregate data, and to look at the analysis and documentation, all in a fully compliant manner. It has the ability to be deployed on the cloud for greater flexibility and security while helping to lower your overall total cost of ownership. And by standardizing the data and procedures across all of these different types of labs, one lab improves lab efficiency and ensures higher quality. The data is captured consistency. You have full audit trails in place for regulatory compliance. And the information is stored centrally for a single source of truth that your scientific colleagues across the world can easily search and find and refer to when needed. It makes it easy to collaborate on new projects, not just by the central data storage, but by the built-in collaboration tools as well. And if you're then creating procedures that are according to industry standards, as you move into the batch processes, tech transfer is simplified as well. Now, a key component of OneLab is our integrated ELN called BIOVIA Scientific Notebook. And it's our new cloud-native ELN, which is based on the 3D Experience platform. It's designed to be really flexible so that you can use it across these different areas in the product development lifecycle, as I was talking about earlier. It also uses that graphical database that I was talking about to help parameterize the knowledge that's created in the ELN, whether it's from free text or other data sources. So then you could better leverage it, such as the use case that we were talking about, but you could expand it into other areas as well. It gives you the ability to consolidate and index data from other ELNs as well. So including BIOVIA's other ELNs and other third-party ones too, so that you can index the data and view it all in a single source in Scientific Notebook. It has integration with formulation design, which is another area of our solution on the 3D Experience platform. So you could have integrated workflows across formulation design and your ELN, and it has synthetic chemistry functionality built into it as well. And so with Scientific Notebook, it really sort of sets you up to have these complete end-to-end lab workflows all across your organization, depending on, irregardless of which scientific domain you're working in. You could use the same type of ELN instead of having to have different ones that are focused for the different areas. And it eases access to the data. It doesn't matter where you are. You could simply search for it in the same type of platform and track projects as they move across that product development lifecycle. It gives you scientists, better access to all of the data instead of hunting for it. And then they still are able to work in the medium that they prefer, whether it's spreadsheets or tables or just typing in free text. It's all available there. And it's mobile friendly, so you could do so on your laptop, desktop, or on a mobile device as well. And it has support for open APIs as well, so that you can integrate it with other solutions. You can pass information back and forth as you need to, and integrate it with other process-based models as well. So Scientific Notebook brings a wide variety of quickly achievable and specific benefits to an organization that implements it in their R&D labs and processes. You can reduce your costs by between 15 and 25% just by eliminating the repetition of experiments that have been already performed. So things that another colleague has done at some point, but maybe you wouldn't be able to otherwise find it easily, or if you don't know where the work is stored or what it's called. If you can't find a reference point to it, you may not be able to find it, or maybe you're not aware of it at all. I talked to one customer last year that said the lifetime of data in their labs is about 18 months, because after that, they don't really trust it as, if you haven't leveraged it in a project at that point, they don't have great trust for how it was captured and all of the context around it. And so by having that source of truth in one place where you could easily reference the original experiment in its native format, it increases the trust and the ability to leverage that experiment down the road. So it takes, between 40 and 50% less time to search for that experimental data as well. So it makes it much faster to find it and share it with others. You could also reduce the time spent creating experiments by 25 to 30%, especially those that are common or repeated in a series. You could duplicate the templates and simply change the relevant observations in that section. Scientists spend much less time creating comprehensive reports, since the data is easy to compile, and the reports can also be created with templates. And so overall, this easily leads to improved productivity throughout the organization's R&D labs. OneLab has, a number of these different capabilities that can be leveraged by an organization. And it enables the key roles in the lab to transform different parts of the lab workflows, as I talked about, the different sort of categories of LIMS and LES and ELN. But the benefit with OneLab is that it could be configured to meet a customer's unique needs, and then can change over time to address different areas as the needs arise or as business priorities change. So smaller companies might only be interested in adopting an ELN or upgrading the one they have, but that becomes the entry point for a larger digital transformation down the road. Or if you have a LIMS in place and you're looking to upgrade from an older legacy version, you could adopt OneLab for information management, but then there's procedure execution capabilities that go along with that. And you could tie those two areas together for more integrated workflows and better data integrity. Or maybe you're just managing chemical inventory in a scratch-built application or in Excel. And if you adopt a cloud-based inventory management solution, that could be a game changer too. But the real value multiplier of OneLab is if you deploy it in one area, then you can expand its capabilities over time. So not only does that tailor your deployment to meet specific needs at one point, but it enables you to leverage that investment and further expand the unified ecosystem. Excuse me, driving both the horizontal and vertical integration throughout your organization for a true connected digital lab. So that's going to end things for me. That's all the slides that I have. And so I'm going to turn things back over and we'll do a sort of live Q&A now.
Great. Thank you, Stephen, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. We'll answer as many of your questions as we have time for. Now let's get started. Our first question here is, can you track chemical inventory in the ELN?
Sure. So we currently have what we call material smart text. So as you're typing information into the ELN, it sort of automatically recognizes things that have been created before as material objects. And so you can link those materials to material tables for experiments and procedures, and for the formulations integration as well. Within OneLab, we have materials inventory tracking as well, along with safety data sheet management and local compliance regulation tracking. And so we're working to finalize that integration with Scientific Notebook as well this year.
Thank you. And then we can't see you yet if you want to jump back on camera.
Yes, sure.
Perfect. Thank you for that. Another question we have here is, where do you see the patenting process in this framework?
So that's an interesting question. I think that it's really important. Basically, you're leveraging all of the information that's in the ELN, right? When you're starting to file a patent, and it really depends on how fast you can get to a point where you have all the information ready to file a patent. And so I think that is something that comes in that sort of early stage research as you start to move from there into process development. You've got everything packaged up and ready to satisfy what you need for a patent. So it's really important to be able to gather all of that quickly from both the ELN and any other relevant data sources, and also publish that in a structured way. So that's something that's important, to have all of that information easily accessible, easy to be collated together and packaged up and written into a patent. So that's sort of where I see it fitting together there.
Thank you. Next question here is your knowledge graph a proprietary data standard?
Sure. So, I mean, really, the knowledge graph, it's a technology for defining the relationships between the pieces of metadata. So the taxonomies and ontologies that you apply there are really flexible. And we normally align those with things like the allotrope data format, or whatever ontology our customers want to utilize. So it's not a proprietary data standard. It's something that uses sort of the different pre-competitive alliance-defined data standards. Is very flexible to exactly what a customer wants to utilize.
Thank you for that. Another question here is, users are often resistant to changing systems. How do you address that?
Right. So it's often the devil, right? It's you might have pain points in your current solutions, but you're used to it and you understand which buttons to click. You know how to move through a process as quickly as possible. And so if you're sort of trying to work with users to get them to adopt a different standard because you see the business benefits to moving to something that's more integrated or streamlined, or for whatever your impetus is from moving on from the solutions you have in place, I think it's really important to sort of lay out the plans in a well-defined manner up front, and to identify the use cases and the pain points that your users have in their day-to-day lives and how you're going to address that. And so that's really about getting those right people involved early in the process, and picking off the specific things that you can tackle right at the beginning, because sometimes those meetings turn into, well, we've got 100 different pain points, so we can't deal with all of them. So what are the ones we're going to start with, and show the benefit there, get users used to using the system and how it makes their lives easier in those specific ways. And then they'll probably start to realize how they can leverage it in other ways too, and then start to prioritize the different use cases they want to tackle next.
Thank you. And we'll end with this one here. Someone is asking, do you have API for LIMS integration general, or do you work with shared data views or files?
Yeah, so I know we do have APIs in place for the LIMS in OneLab. And so I know that we can integrate with different systems that way. That's sort of one of the access points for it. Technical implementation of the APIs is not my strong suit, but I know that it's something that we support, and our tech sales people are more than capable of working with different integrations as necessary.
Great. Thank you. Thank you again, Stephen, for your time today and your important research. We'd also like to thank Labroots and our sponsor, BIOVIA, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we do not have time for, or those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you.